Hey guys, and welcome. So we're going to be doing the Edexcel Biology B AS level exam recap, and we are doing Carbohydrates 1. So carbo means the carbon and hydrate means water, and you can see the elements of CH2O are within the formula for carbohydrates. So we've got types of saccharides, which are sugar units. So saccharides refers to sugar units, and they make up the carbohydrates. So monosaccharide has 1 unit, and disaccharide has 2 units, and polysaccharides have many units. So do not use the word sugar in the exam, because sugar just means everyday sugar. Right. Glucose is C6H12O6, and it is a monosaccharide. It is a hexose sugar, so it has 6 carbons, and hexose sugars are ring structure. The C atoms, or carbon atoms, are at a corner, and so are not shown but are numbered, so here they are. And it is a 3D molecule. So alpha glucose is with the OH group at the bottom of carbon 1, and the hydrogen is above, whereas in beta glucose the hydroxide group is on top of the carbon 1 atom and hydrogen is on the bottom. Right, ribose is a pentose sugar, so it has 5 carbon atoms and it is present in DNA. Here it is. And right, now disaccharides. So disaccharides are made up of 2 monosaccharides, and they join together by a condensation reaction where a water molecule is lost. And they join at carbon atoms 1 and 4, and so therefore the bond is known as a alpha, because it is through alpha glucose, 1,4 glycosidic bond, because it's an alpha glucose, and 2 glucose, 2 sugar units bond together to make a glycosidic bond through a condensation reaction, and the 1 and 4 comes from the carbon atoms which the bonds are made at. Right, so a disaccharide is split by adding water, so it's the opposite of condensation, so it's hydrolysis, breaking with water, and that breaks the glycosidic bond. And examples of disaccharides will be maltose, which is 2 alpha glucoses, sucrose, which is a glucose and a fructose, and this is just another monosaccharide. Lactose, which is found in milk, is through beta glucose, but saying glucose is enough to get you the mark, and galactose. And so therefore the bond is a 1 beta 1,4 glycosidic bond, but saying glycosidic bond is enough to get you the mark. You don't need to learn these structures, although the most they'll ever ask would be to draw a maltose, which is 2 alpha glucose joined together. Okay? And now going through the question, a compound with the chemical formula C12H22O11 could be maltose. Why? Because this is the double of 2 glucoses. So if you add 2 glucoses together, which have a formula of C6H12O6, they will give you C12H22O11, right? But actually, if you look at it, this is less. If you double it, you should be getting 24 C12 and O12. But why? Remember that water is lost. So you have to remove a H2O, which gives you C12H22 and O11. That is crucial to getting the second mark if that question is asked in the exam. And now try this. Give 2 differences between the structure of monosaccharides and disaccharides. While a monosaccharide is 1 sugar unit and disaccharides are 2 sugar units, therefore disaccharides will have a glycosidic bond, whereas monosaccharides won't. And that's 2 differences. And that's it.